Hey Vsauce, Annie here. Imagine a person, like a little random person. You are related to them by 99.9%. Well, genomically speaking, of course. That 0.1% makes you you. However, that 0.1% equals to 3 million nucleotide base pairs, which dictate how tall you are, the color of your eyes, and your risk of getting heart disease and all those other stuff. But what exactly is the other 99.9%? Today I'll be talking about the Human Genome Project. It is a worldwide project that was initiated in 1988 by the US Department of Energy, and which was later adopted as a joint effort with the National Institutes of Health. The Human Genome Project was led by James Watson, this guy over here. The project was first funded by the Congress in the US, which then attracted many major partnerships and contributors from all around the world. These countries consisted of the UK, Japan, France, Germany, and China. As a result, an international and collaborative research program was formed. But what was the initial goal of the project? Why did it go so worldwide? Well, as written in my script right here, their main goal was to develop a high-quality reference genome sequence, which also included the identification of all human genes. They also had minor goals, which consisted of training future scientists in genomics, aiding genome sequences in other organisms, and also providing a resource for studying human variation. Now, before we go on to the method used by the Human Genome Project, it is important to understand how big our molecular genome is. Let's make an analogy. The bases, ATGC, are words in a book, and this book is your genome, which is what basically makes you. As any other book, this book also has chapters, which represents the genes in our genome. However, we have an estimated total of 30,000 genes, so this makes 30,000 chapters in our book. Now, genome sequencing, the goal of the Human Genome Project, is the reading of all the base pairs in the genome, which is basically reading all the letters in our book. In fact, printing our genome sequence on paper would equal to about 262,000 pages, which is like 175 large books to be exact. There are so many nucleotide bases, so it is clearly impossible to read the whole human genome. Or is it? The process of how the Human Genome Project read all these bases is quite complex, so I will go through it with our book analogy. Let's say you were one of the volunteers that gave a blood sample to this research project in 1988. Scientists would take out a DNA sample from your blood, which equates to our book. Indeed, the genome is incredibly long, but it is approximately 8 to 10 molecules wide. As a result, scientists randomly divide your genome into smaller, more manageable fragments by using restriction enzymes, like scissors cutting up your imaginary book into thinner sections. As this process becomes more complex, we have to leave our book analogy for a moment and focus on how the sequencing was actually possible at such molecular level. The answer is bacterial artificial chromosomes, or BAC for short. Back to our cut-up genome fragments, they are then separated, and each one is inserted into bacterial cells. Being in the bacterium that grows and replicates, the human DNA piece becomes part of the bacterium and is cloned numerous times. Hence, a BAC clone is developed. But wait, if it merges into the bacterial DNA, how can the human DNA be seen? Well, our genome fragments are actually much larger and visible under microscopy, just like this shadow behind me. And that helps differentiate the bacterial DNA from the human DNA, and the bacterial DNA is much smaller than ours. Then, our BAC clones are cut even further into subclones in order to carry out a thing called sequence reaction. This incredible reaction requires our subclones, primers, DNA polymerase, and free bases that house specific fluorescent dye, which indicates A, T, G, or C. As the primer initiates the replication process, the colored bases pair with the complementary strands of the subclone. The colored pattern made by this reaction is then processed and analyzed by the sequencer, which is a machine that assembles the developed sequences by identifying genome fragments that were identical to another previously read sequence. And by that, they overlap those regions with its twin, resulting in a stitched sequence. After repeating such actions numerous times, the sequencer eventually develops the complete human genome. Finally, in 2003, the initial goal of reading all the letters in our book was completed. Okay, and? It's just an order of all the nucleotide bases in a human organism. What's the big deal? Well, let me tell you, it's much more than that. After releasing the high-quality reference sequence on the internet in April 2003, enormous amounts of research and discoveries began all around the world, which were all using the product of the Human Genome Project as a reliable biological resource. Great examples of these research projects are the MSSNG project and the study of CRISPR. The Genome Project has also played a role in the control of SARS epidemic in 2003. Not only did advances in the discoveries of science and medicine grow, but the lab method of genome evaluation also drastically evolved as scientists, ranging from physicians and engineers to bioinformatics and biochemists, began to collaborate and develop the most efficient ways of incorporating dry science into research, especially in genome sequencing. As a matter of fact, a simple genetic test from commercial labs, such as 23andMe or Ancestry.com, will now only take 2 to 4 weeks to sequence. On the other hand, clinical testing and research centers will take more than 4 weeks to fully analyze the given genome, due to serious circumstances. It is vital to understand that these faster rates of gene analysis are caused by the implication of dry science and wet science. Wet science will always be somewhat part of genome sequencing due to its involvement in sequence methods such as PCR, BAC, and gel electrophoresis. Yet with the introduction of the sequencer and other groundbreaking technologies, they now work with the results of the wet science, and that produces much more accurate results in a short period of time. Perhaps in the future, genome sequencing will only require one computing system, which would make genetic research discard wet science. But for now, it is a beautiful scientific collaboration between wet and dry science. Now, remember that MSSNG project that I mentioned? Well, a Canadian geneticist, this guy over here, Stephen Scherer, initiated this MSSNG project with the collaboration of Autism Speaks and Google's cloud platform. For a huge time of his career, he wanted to solve the enigma of autism spectrum disorders, ASD for short, which this project made possible. Starting from 2015, the Sick Kids research team analyzed genome sequences and other key clinical data of volunteering individuals with ASD, and also their families, all across Canada. Their goal was to sequence 10,000 genomes and provide such information on Google's cloud platform for researchers that are studying the copy number variation evident in ASDs. The project achieved its goal in June of 2019, yet the cloud platform is still being added DNA sequences to this day. Before I go on, remember that copy number variation study I've mentioned? Well, actually, there's another term for that. Copy number variation, or CNV for short, refers to instances when the numbers of copies of a particular gene varies from one person to another. However, also defined as a CNV, copy number alterations, or CNAs for short, refers to the alteration of the genes, such as deletion, duplication, amplification, and homozygous deletion. Understanding and studying genes that control brain development are vital when it comes to the diagnosis of ASD. But why is this important to study and recognize? Why is it connected to ASD? Understanding where brain development genes are located and how CNAs impact them is a vital step towards earlier diagnosis of ASD through genetic testing. Soon enough, without looking at the physical symptoms of a newborn child, a genetic test can reveal if they have ASD or any other serious genetic disease. Important to remember, the earlier the diagnosis, the earlier the treatment the individual gets, which results in better outcomes. By now, you might be thinking that this is a big deal. I mean, the whole world does, which raises so many conversations considering society, environment, and medical technologies. On the societal level, genomic research plays a role in the identity of people if we're talking about concepts such as family, culture, and ethnicity, since genetic tests are available to trace back into your family history through genetics. However, genetic tests used for research or clinical purposes can be nerve-wracking to some, since serious genetic diseases being revealed early can ruin future plans and relationships. But how about privacy? Where would your genomic information be stored? And who would own it? To prevent such problems, bioethicists at ELCI, which is part of the Human Genome Project, make sure to inform you with all the information considering where and how your genomic information will be used and stored before you do the genetic test, which is called informed consent. Although we have already mentioned many advances regarding scientific and medical technologies earlier in the video, it is important to point out that the field of pharmacogenomics and gene therapy are greatly benefited. Using results from human genomic research, medication can now be developed specifically for an individual. Instead of the one-size-fits-all scheme, scientists can focus on reducing side effects of drugs and focusing on more important symptoms, such as the ones that are caused in ASD. Gene therapy can also be seen to be improved by genomic research as the CRISPR study is rising, which I personally recommend reading about more. Lastly, on the environmental level, sequencing microbiotics can develop various types of sources, such as safe environmental remediation and carbon sequestration. Genomic research also played a huge part in evolution studies, especially when it came to human comparisons. And, as you might tell, biotechnology on crops and livestock has started to rise due to genome sequencing. Feel free to add any questions or comments in the chat below and I'll physically answer them. Now, as always, thanks for watching.